Good evening, folks. Welcome to our fourth Seeds of Knowledge webinar, held on the 4th, the last session of 2024. We'll be back in the new year with 2 more sessions to finish off the program. I'd like to begin by acknowledging the traditional owners of the land from which we join our webinar today. Our guest speaker, Debbie, is joining us from Wurundjeri country, and I'm joining from Wadawurrung country. The seed bank itself is located on Djadja country. We pay our respects to elders past and present from these groups, as well as the owners of the country on which we all conduct our field work. A big thank you to Bull Oak and Northern Grampians Landcare Network for funding this program. We truly couldn't do it without their continued support and shared vision. It is my pleasure to introduce Dr. Debbie Reynolds from Trust for Nature this evening. Thank you, and I'll pass on over to you now.
Okay, hi all. Good evening. Today I'm going to give you an overview of a pretty amazing little sub-shrub that I've had the pleasure of working with over the last, since 2008. So it's been about 16 years. It's only found in the clay soils of Victoria. Its real name, as you can see, is quite long. Pimelea spinescens, subsp spinescens, or spiny rice flower, or plains rice flower are its other common names. Okay, are we moving? Yes, we are moving. Okay. Spiny rice flower is found predominantly in the natural temperate grasslands of Victoria's volcanic plains, but also the black clay soils above the Great Dividing Range. I hope you can see my little pointer there. So Terrick and around those areas there. This is quite an old map that it shows the distribution of probably where it used to exist. The sort of light beige areas, sorry, the darker beige areas are where it probably should exist. The green is where it definitely should exist, and the red is where it only exists now. So, and you can see a lot of linear ones, which are often roads and rails. And both its ecological community are listed federally on the Environmental Protection and Biodiversity Conservation Act of 1999 as critically endangered, and are also listed as threatened on the state's Flora and Fauna Guarantee Act of 1998. Unless of 1% to bandied around a lot of the volcanic plains exist in its natural state still. The recovery plan has just recently, as in March this year, been updated. That's been something that I've worked on since I started at Trust for Nature in 2011. So it's been an extremely long journey, about 13 years, but we finally have a new plan, and that's found on the federal website. Everyone can access that, and it pretty much has everything that we've learnt over the last 13 years is now in there and updated, and I'll talk a little bit more as we go on with that. I also want to show you there is a subspecies pubiflora. So Pimelea spinescens subspecies pubiflora, and that's only found in the Wimmera area. We call it the Wimmera rice flower, is the other common name for it. It was thought to be extinct with no records since 1901 found, but in early 2005, 2 sites were found, and that's a bit clearer there. One was in Natimuck and one was in Minyip. And Natimuck has about 3,500 plants, which unfortunately were partially destroyed by roadworks. That's how we sort of found them, because people went out and had a look, and Minyip has about 191 plants, and that's right in the middle of town in a common, which has she oaks and bull oaks, whereas the lake one actually has a grassland environment. So it's quite different, the 2 grasslands we've found, and since then we found 1 plant in Kalkilli East at the side of the road, and we've also found some more plants up in Salisbury, which is right on the edge of the national park up there. So there's a few more plants obviously that can be found in populations, which is pretty exciting considering the species is extremely rare. And we have germinated seed, which is really cool, and collected seed. And so this is the latest derived map that I got, and it's in the recovery plan, this particular map. So that shows you where the dark pink is where we think all the grasslands and the clay soils are, and when things actually should be. Okay, so the 10-year recovery plan, which is pretty exciting to finally get one. It's only good for 10 years. It took us 13 years to do, so I'm not sure how quickly we'll get to the next one. We have discovered, we think, that a minimum viable population for Pimelea spinescens is about 20, and we would hope that they would be evenly divided between the genders, so male and female, which I'll talk about a bit more. And the same threats pretty much exist: fragmentation of habitat by clearing, competition from both native and exotics, changes in fire regimes, drought, grazing. So we're well aware of the dependence on periodic biomass removal for germination, and the objectives are to maintain positive trends of occupancy where we know they actually are, identify and manage all populations from the key threats, and maintain and enhance community and institutional support, which I think is extremely important, is communities being aware of their populations. And what is becoming apparent is the increased global warming stress that's having on sites close to Melbourne, and people live in rain shadows, so they're struggling with their genetics. Okay, this is the actions from the recovery plan. The cost we worked out, all recovery plans have a cost, 4.1 million. Interesting, it's not funded, obviously, but that's the approximate cost we think it would be to actually run a recovery plan to look after the species. So we think we should protect and manage all populations, insecure populations that we have, increase its knowledge and its ecology, and incorporate them into a management adaptive plans, and increasing community awareness and involvement of land support and managers and community. We also, I think I'm going through a little bit more, the statewide population project, which was also completed in the last few years. Over COVID, we were able to get back out and see some of the populations, and we now know exactly what we do sort of have out there, and it's sort of changed a little bit. We thought that there was a lot less in conservation areas, but we now have about 15% in conservation areas. So that's an increase from 5% from when we first did the recovery plan. There's some in utilities, and private land has 20%. So that's a really important little pocket that we realise that people actually have it on their properties, but most of them on the road and rail corridors, which is what we always knew. And also the sites have increased. So we now have about 400 sites, whereas before we only had, I think, about 220, and the population has significantly increased. But we do know that the population sizes are decreasing over time by the research that I've done, which I think I'll go into a little bit later in the slides. So Pimelea, let's just do a bit of a genus overview. There's about 150 species worldwide, 110 of them are endemic to Australia. They're also found in New Zealand, Timor and New Guinea, and 18 of them are considered rare or threatened. And that's the common one that a lot of people will buy at a nursery, Pimelea ferruginea, which is found in Western Australia. It's Pimelea presii, and then we have a few other Pimeleas that I took some pretty pictures of when I was in Western Australia, and that's Curviflora. The grasslands have 4 species of Pimeleas. There's Curviflora, Glauca, Humulus, and spinescens. And I think they're probably sequentially flowering. So spinescens first, followed by Glauca, then Curviflora. Sorry. Yeah, and I'm not sure about Humulus is in between because I don't see that as often. And of the there's also 36 that are endemic to New Zealand and not found here. Both the Pimelea species that we have are endemic to Victoria only. They're not found anywhere else in the world, and they have pointy stems, spinescent stems. They don't have spines, but they have pointy stems. And we think that's probably an evolution from being eaten, just to give an animal, because they're quite palatable. A lot of Pimeleas are actually poisonous. If you Google Pimelea, you'll come up with Pimeleas in the outback that actually unfortunately poison stock, and a lot of people get quite upset about that. And there's a very good picture of a pointy stem, and if you're an animal you wouldn't really like to bite into that, and they can sort of poke you when they're quite pointy like that, but when they're freshly burnt they don't develop those pointy stems. And Pubiflora, the distinguishing features of it is it has hairy flowers and hairy seed pods, as you can see quite clearly in that picture. And whereas spinescens has quite smooth, which I'll show you a picture of coming up. And also Pubiflora is a slightly larger plant. It's also a bit more yellow in flowers. And there's a picture of it at that's at Lake Road when I went out there and looked at it out there. As you can see, very yellow. Whereas Pimelea spinescens has more of a sort of creamy flower usually. I wouldn't call it white and sort of yellow, it's more creamy sort of colour. This is Pimelea spinescens. They're dioecious, as are all Pimelea species, I'm pretty sure. Pimelea spinescens has male fans. There we go. There's the male on the right of the slide. The male has the pollen, which are the obvious anthers with little bits of pollen on it. And then the female has her central style. And she does have anthers, but they don't have the pollen on it. And the Pimelea is extremely flexible. We have hermaphrodites, so we have plants that also produce pollen and produce seed. It's a sub-shrub, so it's quite small, only grows to about 30 centimetres in height, but it can spread out and become very prostrate, or it can be quite upright. And there's another example of its pointy stems. Okay, insects, all the insects you find on Pimelea. When I first did my research, we weren't sure if that was a problem, that they weren't getting pollinated, because we wanted to know why don't we get more of these plants in the environment. So I went out and took lots and lots of pictures of lots and lots of flies. We have over 1,000 fly species in Australia, and a lot of them seem to love Pimelea. I like to think of it a little bit as a rainforest shrub in a grassland, because it's always in that grassland and it's covered in life. This picture has a caterpillar, it has a little golden beetle to the left there. There's 2 spiders, there's a black spider at the back there, and there's a grey spider over in the bottom right-hand corner. You often just find little things all over it. The spiders are predatory. You do see ants, they can come and go, and I have seen them carrying the seed, but I don't think that's an issue. I think if anything, they're just spreading the seed around, which is great. Lots of flies. I have seen a moth, but I don't think that's normal to see a moth. And I've definitely seen a bee, a feral bee on them as well. Don't tend to see the native bees, I have to say. There is a tiny little bit of nectar in each little flower, just very, very small amount. And so they have to visit a lot of flowers to get that, because as we know, they're quite small. And this is what we sometimes see, which is quite interesting. I was looking at this fly and couldn't understand why it wasn't moving. And when I looked a bit closer, I realised that the spider had it. So I call these the Pimelea spiders, because if you look at their abdomen, they have a very clear sort of Pimelea flower sort of shape, and that's the green sort of form with the yellow abdomen. And then we have a few different other forms. There's the white form, and then there's the darker form as well. And I found them in Pimeleas all over the state when I went looking. The seeds. So everyone's interested in. The seeds are a droopy seed. It's a winter active species, so it flowers from April through to August, bearing, as I said, white to cream flowers. They can look a little bit yellow, but much more likely to be white to cream. Many hours are spent in this field looking at the insects. The 3 mm long drupe, and you can see there that there's no hairs on the actual pod of that seed, and it sort of fades to a browner colour. I thought that was sort of normal, and they have a carpus, so you can sort of see the black carpus there as well. The drupe should be like a peach and a fruit, and then the inner bit is like an egg. And I'm not sure if anyone's from Greece here, but Pimelea in Greece means soft, fat and lard. And so the seed itself is an oily seed, and it will float, and it definitely will, when you squash it in a paper bag, you can see if it's viable or not. It'll leave a little oily stain. And if you squash it and the black carpus breaks like an eggshell, there can be nothing in it, or just a chip, so that actual seed hasn't developed properly. And here's a picture of more insects on them. That's what the seed usually looks like, but this is what I found at a few sites. I've only seen this maybe 4 or 5 times in the whole time I've been looking at this plant, but that's a drupe. That's what the drupe, I think, seed should look like. And this was at a site that had a lot of rainfall during 2010, and it produced this white sort of fruit. And immediately I realised that this, and it's being edible, it's not poisonous. It would have been eaten by birds, it could be eaten by lizards, all sorts of things could eat this food and actually carry it around. And definitely we find a lot of Pimeleas along fence lines, so I'm sure that they go through the bird's gut. And definitely when I've got some experiments that I'll show you later from my PhD, the acid from the stomach and then the base from the small intestine doesn't kill the seed. They can pass through it, and some of my experiments did germinate seed, but it wasn't great, so I didn't get the right sort of ratio, I suppose. Roots. I was fortunate or unfortunate enough to get to dig up a whole lot of roots, because we wanted to know, was it puckering under the ground, and was it a rhizomatous plant? But we definitely didn't find that. It's definitely forming a taproot, and we think it's a claudiform taproot, and it keeps a lot of water in it. We got to dig up 2 sites that were going to be destroyed and try and find out what these roots look like, and every single one had these really quite deep tap roots that went down approximately about 50 to 60 centimetres in the soil. We did have some laterals, as you can see by there, and they went out an awful long way, but not quite as many of them over time. So I think the previous plant that I showed you is actually 1 to 2 years old, and it still has quite a distinctive taproot, and you don't have a lot of laterals in those very early plants. And we went on, you can see that what happens in this plant over time is it gets sucked into the soil. The start of the taproot is often quite a lot lower than the soil, and it has these underground stems, and this is the soil surface. So when the plant's burnt, it loses all its above ground foliage and it comes back from its underground stems. And that's why it can look a little bit rhizomous. And you think, oh, there's a point over here and there's a point over here, but it's still the same plant. But they're all going back to a central taproot. And the reason why the taproot is sinking into the soil over time is because it's in clay, and in summer it's like concrete, and then in winter and when it's softer, it's soft, so the plant's malleable and it can move down. The plant also is quite rubbery itself. The plants, when we had them in the lab, would bend quite happily, move around, and quite rubbery. So I found that when I looked at the roots, there's gelatinous fibres in them which are soaking up water. And there's another example of those nodes under the ground, and you can see how large they are and how they bring back that plant. And then you do have this little weird one that's gone out to the side. No idea why, but it's sent one of its little fingers out over there, and it's still definitely part of the same plant. So underground stems is a common feature of an older plant. This is the plant that we dug up that was probably the largest at that site, and we tried to excavate it as much as possible. And we went, I think it's about 1.5 meters one way and about 1.8 meters the other way, and we were still getting tiny little roots. We couldn't actually get all this plant out of the ground, but we got the crux of it out. And the reason, that's the plant in the lab next day, and the reason why we had that extra bit of taproot is that there was a big rock right there. So the plant had sort of hedged its bets and set a little bit down one way just in case. So that was I've never seen anything like that other than that one plant before. All the other plants had just normal taproots. And this was another very interesting excavation, in that we saw quite clearly that it would just be one plant. But actually, if you look closer, there's 3 little roots going down, and sure enough, there was 3 little roots. This species is very happy to germinate right next to its neighbours, and we often found plants within 10 centimetres of each other, and they would both be quite large and quite happy, and had sent their roots in a slightly different direction into the clay, so they're not competing with each other for nutrients and for water. They're quite happy. And this is what cutting grown will do. It won't grow that taproot, so that's why I don't advocate growing. You can grow them for cuttings, absolutely, and if you were going to put them into your garden or in a nursery situation, sure, but they pretty much 100% have failed every time we've planted them into the field, and certainly you could try and grow them for seed, but they just don't grow the right roots. So that was a 5-year-old cutting that was in a nursery and had been in a pot for quite a long time, obviously, but it just didn't grow a definite taproot, so I would not advocate doing it, and as I said, they've all failed when we've done them. That was another site that we dug up. That's we think those plants were a lot younger. They look quite a bit different in their root structures, in that they were a little bit fresher and younger looking. They still had these little underground areas where you can see the soil. They've sunk into the soil, even though they're quite small in size. We were hoping to age the plants via their roots. And we did do a little bit of that, but unfortunately it all came back. So we did radiocarbon dating of the oldest root that we thought, and they all came back at 50 years. They didn't actually come back at anything older, but I'm sure that they are much older. It's just that we couldn't get the oldest bit of the plant because it's all burnt off. Genetics, we have done the genetics. We were very fortunate at Trust for Nature that we had some funding, and we decided to put it towards getting the genetics done in the species, because to see, were the plants all mixing? Are they very different? And what can we do to, should we move them around? Should we move seed around? Definitely looking at other grassland species, there's issues. So in 2012, Botanic Gardens did the genetics on it, and she took analysis of about 20 samples across the species' entire range. So a lot of stuff almost was my PhD site. They essentially, from the same gene pool, she found all the area sort of from Melbourne all the way across to the west, but up those Echuca areas, those plants have slightly different genetics. So she suggested that we wouldn't mix them, and they've definitely been separated from the rest of the plants for a significant amount of time. Which sort of makes sense because the Great Dividing Range is quite a big divider. So it was interesting to note that we could mix plants all the way out in the west back to Melbourne. And in hindsight, it's interesting in that the plants in the west are in a lot wetter environment, where the plants around Melbourne are actually in about 4 rain shadows. So they're going to be the species and the plants and the populations that are much more suited to a drying climate, with global climate change happening now. So they're probably our most important plants to conserve and to collect seed and to use them for future, when we could lose some of those other populations out the west, which are much more suited to a wetter environment. It said that Pimelea and spinescens are definitely different. That's what we found. And what's the name? Liz James was quite surprised at how alike they were. So she thinks they're definitely very long-lived and probably seed banks. And I did talk to her about birds, that probably birds are moving them around in the landscape and therefore moving the genetics around as well. So a plant could be taken a fair way away, and also the oily seed, the fact that they could float. So if you had floods, they would carry those seeds a fair way along in the landscape, with the volcanic plains being quite a flat environment. So plants within 50, 60 kilometres were much more genetically alike than we'd expected. So they were quite connected. And so this was the end point that she said that those plants up in those areas up there, and these were the red ones of the population she sampled, we wouldn't be mixing them. And Talbot was an outlier. She wasn't quite sure why, but it was very different to the rest. And then the rest of them from the out of Melbourne all the way around to Stawell and Ararat and a few along the Western Plains were all fairly similar and could be mixed. So that was a very interesting thing to learn, and I'm glad we did it. Okay, the population statewide evaluation that Dr. Megan O'Shea did, she started just before COVID and actually continued to do it throughout COVID, which we were very lucky to get done slowly. So her goal was to go back to as many sites as we know, and as we said, we had about 200 sites that we knew of, but they were all named slightly differently. We didn't know if a lot of them existed. A lot of people hadn't been back to some of them in years. So we wanted to try and visit as many sites as we could and see if we if they were still there and what was the population size of those sites. And she also reached out to DECA and landcare groups and many other people to get their information if they had it, so we didn't have to keep doubling up. So there's 236 sites on the inventory with 9 categories. Now there's 323 in 12 categories, 6 population sizes. 183 sites in total were visited. We've lost some sites, and a lot of them were mostly private urban sites. We're in Metro Melbourne, which sort of makes sense, I suppose. A little bit sad, but it is what it is. So Megan did a great job over 2 years, which was really good. And here's a bit of a breakdown of it. So they've got the private and rural properties and the private urban. There is a few of them around. The public non-linear, which is sort of big reserves and things like that, rail reserves. But again, roadsides are the huge place where they are, and they're so exposed. There's at any moment someone could decide to do a plough break if they want to, and also people have farmers and different people have actually decided to take out the fence and extend their cropping. So we have lost sites over time, especially grassland sites out in the extreme west to just complete loss suddenly. And we don't really know what happened. So these are sites that were assessed in green. So Megan got to as many as she could. And large, we don't have massive large ones. A large population is considered between 1,000 and 6,000 plants. So only one was considered, very large was more than 6,000, and large was 1,000 to 6,000, and medium is 500 to 1,000. Medium low is 101 to 500, and then small is between 11 and 100. So the main populations we have in that 11 to 100, and very small is 10 plants or fewer. So we've got those big sites that are in those very small populations, and again, 10 plants or fewer we think is not actually a viable population, and that's a big problem. And we definitely, the trigger for EBBC conditions and having to do something is less than 5 plants. So we're definitely losing plants over time, but they are getting moved, which is really good to see. This is the distribution of sites across local government areas. I sort of throw that in there. So we've got a lot around Melbourne. Brimbank has a lot. Hobson's Bay has quite a few. They tend to be salty adapted and down in different areas around Altona. Melton has quite a few. Wyndham, Campaspe, they're sort of spread quite evenly. Corangamite, Golden Plains. They're spread across 24 different councils, but I think those are the main bigger ones. I think Metropolitan Melbourne accounts for 39% of all sites, and 5% of the other LGAs have them, of the population sizes. So of the achievements that this actually came out with, it's got lots of sites visited, so nearly 60% of the population has been visited, and we're going to try and go back. We've got a database now that's been created. It also was added into the recovery plan, so that's now been updated to reflect exactly what we know. The VBA records have all been updated as well. So when you go into VBA, which you can get most people can get in there, just normal public, so it's available to the public, you can have a look. Each site's 3 to 5 plans were tagged. It's informed the recovery plan, as I said. We now, 50% of sites that we didn't know anything about, that was a target for Megan, and now know. We know what population sizes and whether they exist or not. And we have future recommendations. There's a report that's been written by Megan, and we have plans for how to improve our knowledge moving forward, and also talking about monitoring moving forward. Megan did a little bit of monitoring and data overlay to see how we could do it. So that's been enhanced, which is a great thing. The environmental influences, Pimelea, obviously fire, grasslands, evolution, development of spinescent branches, could be adaptation to put off herbivores. So we've got the adaptations of the spiny rice flower, and in those areas like this, we can see that you can't find Pimelea. It's extremely difficult to find. It's a very cryptic species, as are all our grassland species, hence the earless dragon that we've just found, which apparently has been around, but we just haven't seen it for a very, very long time. Leglesses is another one. They're very small and tiny, and golden sunmoth, all these little tiny, all these animals and flora and fauna are very cryptic. They hide and you can't find them. They've got underground stems, small leaves. They're very grey, green and foliar cover, so they can actually adapt and last in an environment. That's a grassland after it's been burnt, and you can barely see the plants. They're a stress tolerator. They live in an extremely hot environment, which is only going to get hotter. I tried to do some stuff in summer and I was horrified. There's no cover, there's no shade. It's extremely hot, and I don't know how any of these species survive, but it's there still photosynthesizing and still existing in these landscapes. That's our pointy stems there. The plant's growing back from a not quite burnt, and that's an example of what I call a pom-pom plant. That's why we advocate to do a burn. If you want to know how many Pimelea spinescens plants you have, you do a burn in that area, and this is how they will grow back after about 6 months. They are quite clearly identifiable. They pop up. You can easily count them. There's no missing plants. You tend to count them very well, and they tend to be quite prominent in the landscape as a little pom-pom. The other sad thing about the plant at this stage is it's extremely tasty, and there's no spinescent stems, so it's a bit vulnerable to being eaten at that stage. Don't put your stock in there. So the major issues of Pimelea, we sort of worked through that before. Lots of large decrease in the numbers and size. It's very fragmented. Recruitment was rarely reported. This is leading into my PhD, why I did it. And it's not 5%, there's more, so that's a little bit out of date, sorry. But road were albus cemeteries, and there's a lack of management. So my PhD, we learnt, I wanted to identify what is the issue, basically. So I'll go through that stuff. 16 populations I looked at across the entire volcanic plains, and I had study sites, and we did a population assessments. We also did sex ratios, whether they were the issue. We set up permanent quadrats, individuals are marked, 3 to 5 people, and we also collected seeds. So that was where I went back to. These are my sites, so I collected from different sites all throughout. So we captured a broad spectrum of geography of where the plants were, and also sizes, plant sizes. So I had to do some measures of reproductive success for the species. So we collected seeds. So we looked at production, seed production. So the annual quality and quantity is quite important. We looked at viability, and we also did germinability in the laboratory. And we also looked at how many germinants I found actually in the landscape between 2009 to 2010 was when I did my original research. So occurrence over that time, whether there was germinants at all and whether they survived through a year or so. And no research had been done on the species prior to my PhD, whether I did. Yeah, and germinates, okay, I talked about that bit, and that's how you put the seed bags on. I think I'll go into a tiny bit more depth about that, but we put little stockings on them, and that's a picture of what the oily seed stain looks like when you put it with the paper bag, because it was the easiest way to clearly identify if it was viable or not. I'd sort of line up 10 seeds and squash them, and I know it's a bit destructive, but it was the easiest way to know. And the tetrazolium that I tried that wasn't conclusive, so that didn't really work. That's an example of the cute little germinants that you get when they do germinate, with their little seed caps on and the little cotyledons popped out. They're very cute. And that's an example of germinants in the field. They put out these little fat succulent first leaves, the cotyledons, and they often have a little red stem. It's quite distinctive too. Sometimes people think little things growing back from the ground are germinants, but they're actually just an extension of growing back from after a burn. So you have to sort of wait, and they definitely are quite distinctive. And that was a lot of them. They often will germinate together, so that's not uncommon either. The PhD results with the pilot study with the germination, I looked at a whole different ways of germinating these species. I thought with the feed science, you'd be interested in that. What we produce at 72 weeks from 14 different treatments in both incubators, from 2,000 seed, I got 236 seedings, so 11.7% of them germinated. 211 seed, or 88% of the seeds that germinated were in the presence of gibberellic acid, and only 12% germinated without it. 45% germinated in the incubator commencing in summer, while 55% germinated in the one that commenced in winter. Gibberellic acid speeds up germination, but there could be another treatment, but we found it was the best. So that's why I think I've got a, oh, no, go back. Sorry. So you can see every single treatment I did was cold storage, heat shock, smoke, and gibberellic acid was working, but also just gibberellic acid alone. So for the experiment for university, we just did gibberellic acid because we knew that would work just as well. And certainly we got some germinants with heat shocks. So that definitely is a factor, and cold shock as well, and smoke treatment a little bit, but certainly wasn't conclusive, and we needed to know what would germinate this speed. And that's the incubators. We had 2 incubators. One I decided in the summer temperatures. I averaged summer temperatures for the volcanic plains and sent through a 12-week cycle of summer, 8 weeks of autumn, and 12 of winter, spring, and so on, just to see what would happen. And sure enough, nothing germinated in the hotter conditions. I think the summer incubator was like 25 degrees during the day. I can't remember, but the winter incubator was 13 degrees during the day and 5 degrees at night. And as soon as and autumn I think was slightly less, and you had a on-off light cycle in these incubators, it literally turned on and turned off after, I think we just did 12 hours or 10 or 12 hours, depending on winter. And definitely once you got into the colder conditions, we started to get these spikes in germination. And you can see in the winter incubator, within sort of 7 weeks, it does take a while, you start to get that germination. They don't tend to germinate, they take a little while. It's normal for it to take sort of 4 to 5 weeks before you get much germination, but then you'll get it. The other interesting thing is they'll just keep germinating in the cool conditions. We've kept incubators in cool conditions for months, and they just keep germinating. And that was the other thing about that winter incubator. As you can see, we had it through summer, and every time we got warmer, the germination rate decreased to nothing during that summer. And then once we went into cold conditions again, we got another germination spike. And this was from exactly the same dishes with exactly the same seed. All I'd done is remove the seeds that were germinating, and I just added water. So it was always kept a bit moist, and we did a gibberellic acid treatment once at the start, and that was it. So the plant's seed will respond later, and that this goes to show that not all viable seed would germinate. So they had a seed dormancy. It's I forget what it is now, I haven't written down, but it's definitely the seed dormancy of the species, and they don't all germinate, which makes sense. And this is the germinations in situ. So we had in 2009, 2010, we had germinants per female plants, and that was the time since biomass reduction. So we had more germinants in the field in sites that had a shorter time that the biomass reduction had occurred. The longer the biomass reduction had, so 5 years, 2 to 5 years post any sort of biomass loss, you had less germinants, way less germinants. So you need to have more biomass losses, and you'll get more germinants in the field, which sort of makes sense. And this is the survival rate. So if you had a better quality, this is with habitat hectare scoring that I did, increased diversity of species, and also had a greater percentage of bare soil and crisp bare soil, less weed cover, and less organic lift out. You definitely got more germinants that survived, which sort of makes sense. And mature plants was another one that we had mature plant mortality in the time that I was looking at them. Half the sites had plant deaths, 4 only had 1, but one study had 9 plants that died, and 54% of the plants were non-flowering. This is actually in a site around Melbourne. And that the mature plant survival was positively associated with a greater percentage of bare soil at a site, just like germinants. So they also need biomass loss to actually survive and do well, so which sort of makes sense as well. They like being in open areas. So seed germination, this is what happens. Seed germinations will go during winter. So if you want to seed your germinants on the ground, the female plant density positively influences germinant density, and there'll be more germinants with shorter periods between biomass reduction. You need more bare soil, less weed cover, and less organic cover is the other thing. Because once you burn, you get rid of all that. Less biomass. I was actually lucky enough to go back to my PhD sites in 2018 and revisit them. This is part of some research I was able to do. Went back to the cod rats and caught another snapshot of the population. 2 sites had no mature plants die in the last 8 years. 11 sites had new plants, ranging from 1 plant to 31 plants, with 3 sites having no new plants found within the cod rats. In the last 8 years, across 14 sites, unfortunately, 2 sites had been damaged, quite significantly damaged, and I couldn't find quite a few of my quad rats, so that was really distressing. I only had 16 sites. There was an average loss of 2.2 plants per year and a growth of 10.8 plants. So we actually are losing plants across sites over time. And you can see sort of the breakdown of these sites a little bit, whether we had deaths, we sort of deaths at a lot of them, and whether we had new plants. And we did have new plants, but what we did find was the growth rate was better, greater than 1 at some of these sites. There seems to be a lot of populations that were in the middle and hadn't changed at all, so we're pretty much even keel, but it sort of balanced each other out. You've got plants, sites that are doing well, and the sites that were doing well were getting biomass reduction. So they're actively managed for burning. Ararat had a recent burn. Mount Mercer is routinely burnt as it's a strategic fire break. Pitfield Chris is definitely burnt. Glengower is another strategic fire break. But some of these other ones hadn't been burnt in a very long time. So they're definitely regressing, that I found. I forgot to do more research around Melbourne. This is at a site that we were trying to collect seed. And we wanted to see if burning, mowing, or doing nothing, what that would do to seed production. So yes, I think I've explained that. It was 2 areas very close to each other. There was the results were that there's no significant difference between the sites or years. After 2 treatment timings, burning significantly affected the flowering and the seed production. So we had collected seed at this site in 2012, and we got something like, 400 seed from 22 different individuals. In 2013, we collected 758 seed from 19 individuals, so less plants were producing seed. And then in 2014, post the burn, I think it was I can't remember if it was an autumn or spring burn, but we got 6,216 seed from 59 individuals, so way more individuals actually produced seed. In 2015, 892 seed was collected from 50 individuals, so slightly less, but it seemed to maintain a much better status quo. And then 2016, 3,000 seed from about 52 individuals. And then we did another burn again. And then in 2017, we got 10,700 seed from 58 different individuals. So almost all the plants that could produce, there was 59 in 2014, 58. So it definitely stimulates the production of production of flowers, which corresponds to seed production. The other interesting thing was it actually increased not just the burn area, but the mow and the control. All plants produced better seed following a burn, and we think it was probably because the burn plants looked amazing and all the pollinators were rushing in to get to them, and they were also incidentally finding the other plants who were doing okay as well. So it was that's sort of what we think, but it was sort of interesting that they definitely produced more seeds. I also got to do some I grew plants, and we got to establish them at different sites. So we have a grassland sites, and we had a plough sown with forwards, which I thought would do better because they do like lush sort of soft soil. First people who came to the farms all said that the soil was very soft. It was friable and soft and beautiful, and that's what I want to do. And a lot of the forward fields aren't like that anymore. So I want to try. So we ploughed a field up, and so of course, and this area had been ploughed for many, many years, so it wasn't any different. And we've also put in mow, burn, and control into those sites, which we didn't get to do so much. And that's the plough site. So we ploughed and sown it into September 2012. This was trying to get Pimelea established back in the field. And we put seeds in it. That was done by Greening Australia. November 2013, the plants finally started coming up. It took a year or so, and we had to do a lot of weeding around the edges, a lot of weeding through it. And then in January 14, we got a lot more species coming up. And it really started to look like a beautiful forward field. The plants did really well in these forward fields. I'll show you in a minute, and that's 2016. It was just stunningly beautiful with lots and lots of different species. We didn't have as many species as I like, and I started to try and add stuff in there, but everything seemed to hang on and do really well in this area. And those plots are where my plants are. So we planted plants into grassland and also in the ploughed wildflower field, and we had sort of results in that we averaged 22% survived in the grassland and 41% survived in the wildflower ploughed field. And over time we had 160 plants alive, 31% of what we'd actually grown. So we grew an awful lot of plants in an incubator. which I think I'll show you, and we got them out in the ground eventually, and we looked after them. We watered these plants for at least 2 years. They need to be watered, otherwise they definitely won't survive. And then after that time, they seem to do fairly well. And that was, there's I think about 4 or 5 plants. We planted them in clusters because they seem to like being next to each other. And you weren't sure whether they were males and females too until they actually flowered. And plants did definitely flower in their first year. They just were small, but they definitely produce flower and can produce seed in their first year. So the plough was the best method. We also planted plants earlier. We did do them later, and then we planted in May. They were much better. They actually were able to establish much better and be ready for the summer period. And in plot 13, we were really excited to see that we got a germinant. So that was the whole thing of the experiment, was what we hoped they would eventually do it on their own. Once you've got enough plants there, they would produce their seed, which I didn't collect. So I never collected seed from the site, and we would get little germinants come up. So that was an exciting moment to find a germinant in my little plot that I didn't have to plant. And that plant survived. That's it in 2018. And hopefully that site's continuing to do that. I haven't been able to get out there for a long time. It's in the Western Glassholm Reserve, but it's still privately owned. So management implications: greater bed soil promoted flowering, allowed greater number of seeds per stem to be produced. Burning section improves the number of seed by chance in all treatment areas. So that's a really interesting thing to do, that even if you only burn a part of your property or a part of an area, it will still benefit the plants that are there. Less grass, better bare soil, significantly improves establishment, and a lot more seed means you have a lot more chance of germination and recruitment, which sort of makes sense, you would think. Seed collecting, you place little bags on in about July, August. I always put them on. You've got your female there producing her seed, and she's got little flowers on her there. And you can see, once you can see little seeds, they're like little clusters of grapes. That's quite common, and you can often see seeds all up and down that stem. When you see that, that's a great stem, and if you can, try and bag up at least 3 stems. You want to try and collect at least 100 seed from a plant if you can, if that plant's large enough to produce that, and usually one of those stems would have up to that amount on it. Showing a few different pictures in August. That's me putting my little stockings with little sort of bread ties on the end of them, and I put little bread ties at the bottom, and they seem to be very robust. We haven't had any real issues. If there's animals caught in them, they just eat their way out and they seem to be fine. I've come back and found moths and ants and all sorts of things, and luckily they seem to come out the top, so they usually seem to keep the seed, and they seem to be lasted quite well. I haven't noticed that many mold issues at all either. And then you move the bags in about October, November, because that's when it's starting to get warm, and the seed will just naturally fall off. You don't want to be picking it off. The seed is quite green for quite a while, and I think the plant's using them for photosynthesizing. So the fact that it grows during winter is a pretty stressful, hard time to get anywhere, and it just takes a lot slower and slower time to get anywhere. So keeping those seed on them is really important, I think, and then they do just naturally drop once it gets warmer. And that's what I use, is my trusty ice cream container, modified, because it can be a bit hard. The seed's quite round and it can sort of ping off in all different directions. So I put the little seed bag in my little side bit, and I've cut a little bit more of a hole, a little bit, and then you open up the bag slowly, let the seed all fall in, and then slowly take your seed off and check, and then pop it straight in a little paper bag. Fold it and fold it and fold it again, because in a grassland it's quite windy, and I usually use little clothesline pegs and peg it to my basket so it doesn't fly out somewhere, because often it's windy in a grassland. Of course, you need site, date, plant number, species, who's collecting it, where you're from. Fold it and fold it quite a few times. Never have enough folding. Example of some of my bags, and they're from all the different sites that I collected from. You don't want to expose it to heat. We kept it in the university. We kept it there. And this is germination. So we store seed during summer. Megan has tried to germinate them a little bit earlier, and they can go earlier. If you put them straight into cold, apparently they will germinate straight away. So that's something we've tried a little bit. But I tended to leave them over the summer period, because that would be normal on the ground. They would lie on the ground during the heat of summer and they wouldn't germinate. But the next year, as of the cool temperatures start to come in, so 1% gibberellic acid, you can buy this at different places. It's a natural plant hormone that overcomes the inhibiting hormones. So this species has inhibiting hormones that make it not germinate, but the gibberellic acid will overcome it and actually allow it to germinate it. And then we place it into the incubator with the winter temperatures. Gibberellic acid is quite sensitive to light, so we put foil around it to protect it. and just use it sparingly. I just use a pipette dish and put it in my Waltham paper. I use a little kitchen and literally from the supermarket, little kitchen sort of paper, and we keep that with moisture with water, and then we just put the Waltham paper is soaked in the gibberellic acid neat, and then popped on top, and all the little seeds are placed around, and we name where it's from, what plant number, how many seeds, and when we did it. And that's a big event when we did that. Many years I did that. Perhaps into our incubator, which isn't a cheap incubator, so 13 degrees during the day and 5 at night. And then I would check them sort of weekly from about the 3rd or 4th week. A lot of people think I'm doing custard tarts. It looks a bit interesting in the incubator. We've got the light as the trays go, that it literally goes on and off. Some of the more fancy incubators can grade up and down, and the temperature does grade up and down to 13 and down to 5, which is pretty good. And then if you're lucky, you'll get some little baby germinants. They're very sweet when they come out. It has a nice strong root normally, and we do get them with mold and funguses and stuff all over them, but I sort of try to scrape some of that off. It doesn't, it can overcome a weak germinate, but not necessarily. And certainly talking to botanic gardens about it, they say they never do any fungicides with any of the stuff. They grow in dirt anyway, so that's quite normal. So we sort of just leave it. And we did find that we have super producers. Some of the female plant that I collected seed from, when we did dishes, we would get a guaranteed about 3/4 of that seed would germinate, whereas other dishes, more likely the average was maybe 3 to 5 seed a dish for sort of 20, 25 seeds. So there's another variability going on with the plant themselves, and I just put them in little tubs initially, and that's what a little germinate looks like. So they do a bolting of their roots to get their roots down into that lower surface in the soil to the moisture as quickly as possible, and really don't have a lot on the top, as you can see, and often they're next to their mothers. We think that in wet years they prefer to be a bit further away, but in dry years they actually need to be closer for the thermotranspiration that their mothers provide. There's often a mum and her family all around her. When you find a cluster, they're probably genetically related, and we did find that a little bit where Liz did the genetics. And there's some little babies with their little roots. They've got a tiny bit on top, but they've definitely got at least a 4 centimetre root coming out the bottom. So I usually try to get them in the ground pretty quickly if I can, because that roots, and whatever you do, don't break that root. That's probably the worst thing that could happen. They need that little tap root, and it's really, really important. And sort of getting them in as early as you can to help them establish in the soil on their own. It's tough going in clay. The planting, I plant in autumn if I can, once the moisture is in the ground. We've got our trusty drill, because often the ground's not as soft as your bread. Definitely get different plants for different genetics if you can. So if you're going to collect seed, collect from at least 10 plants per site, and try and collect across the site if you can. You'll need a permit, of course, if you're going to collect seed, but you can collect on private property without permit, as far as I'm aware. And the FHE permit system's a bit different now, and it's definitely worth having a look at it. It's all new, it's all online. It's not as onerous, I suppose. Group plants no further than 5 metres from other plants. Always try and plant next to plants if you can. I use my trusty spatula, like a cake's blade, to get them out, and I always cut my tubes normally because I'm terrified of breaking that taproot. Get them in the ground, and this is my seed orchard that I did next to the other area of the ploughed field. So we tried to do some genetics and get a seed orchard in the ground to try and look after. This is what we need. If we don't have seed orchards, we won't have species in the future. We put in some pipe underneath. I went and looked at a whole lot of different nurseries and the Arboretum and Euroa and recommended this method. So it was underneath the shade cloth. And then we put the shade cloth over the top of it. Unfortunately, the sort of mounds were huge. I didn't quite expect them to be that big. They should have been a bit smaller, but anyway, we worked with what we had, and we planted them in like that into the shade cloth. And they actually did really well, because I know that shade cloth protects them. And I also put around cages and little baskets with the shade cloth, because they would normally be next to their mothers or other plants, so they would have some protection. It was a very exposed site, so I just want to give them some protection. It wasn't sort of for sun or other things, but this is just to give them a little bit of help to keep going. And we had a water system, we watered them, and we also just went out and watered them as well. And over time, little grow, I collected from I think 17 different sites in the end, and we were slowly adding to them over time, and they did really well. We got to the end, there was nearly 143 plants across the site, 17 different populations with 16 different plants on one site, and the aim was for 30. So we were going to try and build that over time. That's why we have the sort of big gaps there. It did fairly well. The plants grew quite large, and we of course had males or females. You didn't know until they flowered. And the perfume that side was amazing. And we did collect seed for at least three or 4 years, I think. So we had people come out and help with the seed collection days, putting bags on, taking bags off, which was great. The Pimelea recovery team, I asked them to come along. We had different councils and consultants and people come along, which is great. And got quite a bit of seed, which I still have a fair bit of that seed actually, and we had to weed. So the future is possibly there was a dog that was trained for Pimelea to try and find it when we can't find it, and that's a really great thing to do when a lot of the species that is cryptic, the earless dragon as well, has dogs that look for it and find it in the holes. So at least that way when you're trying to look for it at the times where you really can't see it in the landscape, the dogs have more of a chance of doing that. Seed orchards, we need more seed orchards. We really need one above the Great Dividing Range. Would be great to look at those all those sites and actually collect seed and do some seed orchards. And that's what my seed orchard looked like when it was profusely flowering in June 2017, and the smell there was amazing because the Pimelea family is perfume. It's Daphne family, so it has a beautiful perfume and a little tiny smell. And signage, another thing I advocate hugely is please put signs up. It's often the last great effort, not this huge sign obviously, and that's actually out on the old Melbourne Road near Avalon, and they did that for the air show because people were parking on the sides of the road for the air show, and now they've got it cordoned off on little fences and they don't do that there, and that was because of dumping. Roadside dumping down that road was constantly happening, so they pop a sign up. But this is the standard one we sort of recommend if you can have something there, just to, if you've got a council worker or a farmer or someone coming along and they just decide to do something, at least they have a bit of a twig that they really shouldn't be doing it there. And you can't be out there all the time to look at it. It's just the last sort of great bastion, and we definitely think it's worthwhile. And this is a great sign that Whitcliffe Cemetery was done by Aggie Stevenson at Glenelg Hobson's Council, and that's a beautiful roadside there and just has a lot more interpretive with it, which is great. Growing and augmenting sites, that would be great. That's what I'd advocate, and I've definitely done this with people, and I'm more than happy to help people if they have sites and they've got private properties. I'd like to do this more. Collecting seeds on the sides of the road, I have an FFG permit obviously to do this. So Arcade Way in Avondale Heights only had 2 or 3 plants. We planted 10 and 3 survived, but it doubled the population. It was a good start, and that was during COVID. We sort of got to do that, and I'd like to do some more for them. Pioneer Park, they've got translocations, natural, and I've put in about 54 planted, and we still have 17 alive. Well, I think there's a few left today, but we're doing some more plantings into there. So we've augmented that site. And Skipton Common was another interesting one. That has a lot of plants. There's over 1,367 natural, but we've taken different genetics from different areas and put them into that site to actually augment their genetics. And we did it with a local with the schools, with the STEM program with the girls. So that was and they actually go out and monitor those plants over time, which is really great as well. So it was really building for the community and helping everyone understand where your sites are and what you need to do. And I've finally finished. I've got some pretty pictures of Pimeleas because they're the cutest thing ever when they germinate. They get their little seed head with their little cap on them, and then they pop it off and they still have their little seed cap on them and they keep it. I love that. It's like a little hat.
Yeah,
they're wearing their hat.
Cute.
They're very sweet, and it's so exciting to see that. It's like, oh, thank God, we've got a germinate, and there's a few breathe, and there's a classic hermaphrodite. So there we have the male flowers, and we have some seed on the same stems as the same plant. So LGBT is alive and well in the Pimelea genus.
Wonderful.
Yeah, and questions. There you go.
Oh, thank you so much, Deb. That was great. And I have a few questions here, so I hope you don't mind. But
no, that's fine.
Before I launch into it, I just want to say those Pimelea spiders are really cool.
They are very cute. I think they're found on lots of plants in the grasslands.
They're adorable. So you kind of answered one of my questions, but I was just going to ask for people in the audience, if they do find a Pimelea or what they suspect is one of the endangered Pimeleas on their property, do they contact you, and then what does the process look like after that?
Well, yeah, take a picture. There's a few triggering things that, is it winter? Is it flowering? So it's because sometimes glaucas can be mistaken for them, but they tend to grow a bit bigger. They're quite a bit more of a bush. So they're quite distinctive. That's the thing. If you want to see where they flower, but yeah, I've definitely had people send me an email with pictures in it going, I think this is spinescens. So yeah, it'd be great to know and ID it and get someone, there's plenty of other people that can ID them as well, which is great. But it's good to know, and we definitely want to make sure we know where they are. If they're on private properties, add them to the databases, so everything is up to date. And we do, as we said, 20% are on private properties. So if you've got a site that's not overly grazed, I think they will make it there. And they're definitely a species that hangs on. I know Warren Bean has them. They lightly graze different areas, and Terik is another classic that I think had some as well, lightly grazed for many, many years. So they can tolerate it. But it's good to give them a rest, I suppose, and I suppose you want to encourage them. That's the thing. You want to see how many do you have, look after them. Burning is really important too. Then you'll definitely see what you've got.
Yeah.
Grassland species love a burn, and it's always a good thing to do to find out if you've got a bit of a grassland, is just do a little spot burn in an area and just see what comes up, because you might surprise yourself. A lot of stuff is dormant and seeds in the soil for many years. And it would be amazing. You might have orchids that you didn't know you had.
Yeah, I've just had another question come in, so I'll just ask that one quickly. How frequent does watering need to happen north of the divide?
I'm not sure, because I haven't had a lot to do with them above the divide, but the genetics for up there would probably suit it to up there. But I've definitely found they need looking after for a couple of years. If you're growing them and you're planting them into a spot that they've not been there before, you're better off looking after them. If you're planting them next to other Pimeleas, then they're sort of going to do maybe better, because the sites I showed with my seed orchard and also the experimental ones didn't have Pimeleas there, so we weren't even sure if they would live, but they did. And we definitely think there's mycorrhizal stuff going on probably in the roots. And I definitely, when I dug up my plants for the roots, there was actually cocoons in them. There was animals living in the root, like a real hole, like there was a hole but the plant was fine. You would never have noticed. So there's definitely a lot of stuff going on there, and I definitely think mycorrhizal is important. So definitely, I know Megan has said that she's sometimes gone and taken some dirt from under a Pimelea plant and used that with plantings and with the dirt that we use. When I do the plants, we actually use at least a half clay, half potty mix sort of thing. Don't use pure potty mix. They need some resistant to grow, and they're going to grow in clay. So
that was actually going to be one of my other questions, was mycorrhizals, and whether that's why the gibberellic acid has some good results, because that's found in the natural world in microbes and things like that.
Yes.
So yeah,
we don't quite know, and I have talked to Botanic Gardens before about it, but apparently if they use anything with the funguses and the mycorrhizal, there's like a mix of all 5 or something. There's 5 inoculants you can use. We haven't actually done that, but yeah, we're open to learning, and we certainly want people to let us know what they have done. And I've definitely worked with people to germinate them. I worked with the Mount Curong. I forget the name now. Her name is Kate, and she had a little nursery, and she had Pimeleas on sides of roads, and we definitely worked with her in her nursery, and she did end up germinating some plants and getting them out in different places. So putting them back on properties and trying to establish them is really something that would be great in play areas that are secure. That's what we need. That's the problem. They're all mostly on roadsides, which aren't secure. So we sort of need to get them into those other places. Safe. Yeah, it's capture the genetics, not lose them, sort of thing. Even if you collected seed and put them out, we have done some direct seed germination stuff, and it unfortunately hasn't been very successful. I think we've put like 2,000 seeds plus seeds, and we got germinants. We got like 9 germinants. But that was it.
I only laugh because that was going to be my next question, was chances of direct seeding in the future. But
we've definitely tried it, and we'd like to do it, and it's not that we don't want to do it, it's more it just hasn't. And I definitely did some stuff on dirt in the lab, and it didn't tend to work as well for some reason. They liked the light. I think they liked being open. It's sort of interesting.
I was just wondering if you've come across any populations that were all female or all male, and what, I suppose you do some transplanting of the other sex into that population if you find that.
Interestingly, there's one population that I know of that has and you might know Mount Rothwell has
Yeah.
unfortunately only males that we know, and maybe one hermaphrodite. So yeah, you definitely need to we need to put we need to try and get some females in there. Basically, it's not it's not a viable population if you're not producing feed. So I've definitely worked with them a little bit, and I'm going to try and do that again. We're actually in the process at the moment. I collected some seeds along the rail reserves actually in the local area, and hopefully we can work with Donna, their volunteers, to try and with Megan, because that's their thing. It's quite labour intensive to get these germinants, so we sort of need people to help a little bit in that area. Megan's done it for she's done it for consultants, and she's done it for DECA in a few places because they've employed her to do it, because it's extremely labour intensive. It's not for the faint-hearted, and it takes a lot of time. You really need to be, like I did it part of the PhD, and I sort of couldn't let any of the germinants go. I had to pass them on and make sure they had a life, just in case.
Yes.
Felt bad germinating them and not potting them up and giving them to someone.
Yeah, of course, with your little babies.
I know. Yeah.
Mount Rothwell was where I met you with bagging of some of their populations, so that's where our journey started.
Yeah, well, I'm still doing it. I'm trying to do it on roads that we're losing, like as I said, I did along the railway line because of possibly that rail hub was going in around Little River. So we targeted that rail line because that's got lots of Pimeleas on it, and there's definitely the outer metropolitan ring road will happen, and there's quite a few populations or little sites with little plants on them. So this is something that had to happen over the next few years if we can. You need to bag the plants up, you need to get that seed so that you've got that genetic capture them before the plants are lost, sort of thing, or they're moved and they die. You've lost them completely. Like, they will move, a lot of them, but we're realizing that if we don't do the seed collection at the right time, no one ever wants to do that. It's a long drawn out sort of thing, but I'm happy to try and do that a bit more.
And last question before we go, I was just wondering, you mentioned earlier about how a lot of Pimelea species are poisonous. I was actually wondering if there was anything we should also know with any cultural practices with Pimelea. If you're aware of them?
Yeah, not that I'm really aware. It was called bootstrap bush, apparently. I think that was from the early people who came here. The Pimelea glauca stems were so strong, they used them as bootstraps. So it's a very strong fibre, and I've definitely found it's extremely difficult to break them off. You need cutters.
Yeah.
So they're very twiny and tough, and so I'm sure they would have been used as like tying something, or they probably and like attaching something, a weapon, attaching something, maybe they could have been used. So I definitely understood. I'm not aware of food or anything like that necessarily. I've not heard of anything like that. But definitely they were in the landscape, and they could have been used for water. Look, that's the other thing that's even stranger, because they're definitely a water storage vessel.
Yep.
So there's a few different, that's what I was saying, when I dug these things up and we put them in the incubator, we put them in a freezer for quite a while before I did a lot of the testing. And what I noticed was actually they kept growing. We put them in a pitch black fridge, and at 5 degrees or whatever was a fridge, and we just closed the door, and I walked off, and I would come back periodically and do all my measuring and all the rest of it, and I'm going, oh my God, these things are growing. They're actually putting on fresh foliage, and
that's amazing.
what was happening was the roots were the taproot was becoming more wrinkled. So we're using their water that was stored in there. Yeah, and there's a lot of lignin on the edge, so the lignin's holding that water in, keeping it. It's only the sort of ends of their stems, so the ends of their roots, that are bringing up that water. It's quite
amazing
fascinating.
Little plants. Yeah,
they are. They're extremely adapted to that environment, and that's why too, when people have scraped the soil sometimes at sites, they haven't always killed them all, because they've not scraped off their taproot. They've just scraped those stems, and they can cope with that. Yeah, they've grown back.
Well,
so, yes, but they would kill some of them, but not, the older ones. That's the thing. That was my theory. The ones that are deeper in the soil are older, but we still don't know how old they are. That's what's even more annoying. I'd love to know how old they are.
I was going to say, they seem like one of those little plants where the more you learn, the more you realize you don't know about them.
Well, grassland species generally, it's all about the roots. Their roots are absolutely amazing and have been there for a very, 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 very long time, and you can't age them, really. So it's quite sad, and we don't value them then. Do you know what I mean? We
Yeah.
value a tree because you can see, but I think of their
Yes.
taproot as like their trunk. It's a massive trunk under the ground, and we don't sort of see it and think it's important, which is sad.
Yeah, we definitely need to get that out a bit more so
Yeah.
people can see it. Well, thank you for joining us tonight, Deb. We really appreciate it. And there's a lot of comments in the chat saying thank you and thanks for the great presentation. So everyone has really enjoyed it.